Friends, today we will learn about microbiology. Different different topics in microbiology we will discuss in this video. In this video, first of all, introduction. Introduction. Then we will study about scientific development of microbiology. Here we will study about Louis Pasteur, Joseph Lister, Robert Koch, Paul Enrique, and virology. We'll study about virology, immunology, some idea we will discuss in this video. Chemotherapeutic agent also we'll discuss. First, we'll discuss about introduction. What do we mean by microbiology? First of all, microbiology meaning we will discuss. Microbiology is the science of living microorganisms that are only visible under the microscope. Now let's know about medical microbiology. Medical microbiology means it deals with the infections and infectious disease. Medical microbiology deals with the causative agents of infectious disease of man, his reaction to such infections, the ways in which they produce disease are the methods for their diagnosis. The disease is transmitted by contact for man to man was known since biblical times. Viro and Varro and Columella, first century, that is before Christ, postulated that diseases were caused by invisible organisms which they called Animalia minuta. Praco, Fracastorius of Varona proposed a contagium vivum virus, living germs, as a possible cause of infection, infectious disease. One Plencia Francis in 1762 suggested that each disease was caused by a separate agent. Kircher reported finding minute worms in the blood of plaque patients, but it is more likely that he observed only blood cells with the equipment available to him. As microbes are invisible to the unaided eye, definitive knowledge about them had to await till microscopes were developed. The credit for observation and description of bacteria goes to Antony van Leeuwenhoek. He was from 1632 to 1723. A draper from Delft, Holland, whose hobby was grinding lenses. He described three major morphological forms of bacteria, communicated his findings to the Royal Society of London. The significance of these observations was not then realized. In 1678, Robert Hooke developed a compound microscope and confirmed Leeuwenhoek's observations. The Leeuwenhoek, the world of those little animal molecules, represented only a curiosity of nature, and their role in disease was not realized. Their observations lay dormant almost for a century. It was only some 2 centuries later that importance of microorganisms in medicine was recognized. The earliest discovery of pathogenic role of microorganisms probably goes to Agostino Bassi in 1835, who showed that the mus- muscardine disease of silkworms was caused by a fungus. Oliver Wendell Holmes in 1840 and Ignaz Semmelweis in 1846 had independently concluded that Puri, puerperal sepsis was transmitted by the contaminated hands of obstetricians, nurses, and medical students, and this could be prevented by washing hands in antiseptic solutions. Nidam, in 1745, an Irish priest believed in spontaneous generation of microorganisms from the decomposing organic matter. He had shown in his experiments that microbes developed in closed flasks of previously heated putrescible fluids. This view was posed by Spallanzani in 1769, who found no such generation of microorganism after heating, long heating of putrescible fluids. He proved that organic contents of flasks supported the microbial growth when exposed to air. Later on, Pasteur proved conclusively that all forms of life, even microbes, arose only from their like and not de novo. Now let's know about scientific development of microbiology. Scientific development of microbiology was ushered by Louis Pasteur, perfection on microbiological studies by Robert Koch, the introduction of antiseptic surgery by Lord Lister, and contribution of Paul Enrich in chemotherapy. Now let's know about Louis Pasteur. Louis Pasteur, he was from 1822 to 1895. He died in 1895. He was a trained chemist of France. His studies on fermentation led him to take interest in microbiology. He established that fermentation was caused by microbial agents. He further noted that different types of fermentations were associated with different kinds of microorganisms. He is also known as father of microbiology. Now let's know about some points he contributed to microbiology. So, first point is development of methods and techniques of bacteriology. Second point, proved conclusively that all forms of life, even microbes, arose only from their like and not de novo. Pasteur, third point is Pasteur also had to face challenge from Pochet, who was a proponent, proponent of spontaneous generation theory of microbes. Pasteur disproved the view by demonstrating the ubiquity of microorganisms in the air by his experiments performed in the swan-necked flask. Introduction of sterilization techniques and development of steam sterilizer, autoclave, and hot air oven. Studies on anthrax, chicken cholera, and hydrophobia. During studies on rabies, though Pasteur could not isolate any microorganism from man and dog but suggested that the causative agent of rabies was too small to be seen by microscope. Sixth point is live vaccine. He introduced attenuated live vaccine for prophylactic use. An accidental observation that chicken cholera bacillus cultures left for the several weeks lost their pathogenicity but retained their ability to protect the birds against subsequent infection by them led to the concept of attenuation and development of live vaccines. He attenuated the anthrax bacillus by incubation at high temperature, that is 42 degree to 43 degrees centigrade, and proved that inoculation of such bacilli in animals induced specific protection against anthrax. He convincingly demonstrated the protective role of anthrax vaccine in a public experiment, that is 1881, where vaccinated sheep, goats, and cows were challenged with a virulent anthrax bacillus culture. All the vaccinated animals survived while an equal number of unvaccinated control animals died. Pasteur coined the term vaccine for such prophylactic preparations to commemorate the first of such preparations, namely cowpox, employed by Jenner for protection against smallpox. Pasteur's development of a vaccine for hydrophobia marked a milestone in development of immunization in medicine. He obtained the fixed virus of rabies by serial intracerebral passage in rabbits. The rabies vaccine was prepared by drying pieces of spinal cord from rabbits infected with fixed virus. This work was acclaimed throughout the world. The Pasteur Institute Paris was built by public contribution for the preparation of vaccines and for the investigation of infectious diseases. Now let's know about Joseph Lister. He was born in on 1827 that year and died on the year 1912. He was a professor of surgery in Glasgow Royal Infirmary. He applied Pasteur's work and introduced antiseptic techniques in surgery in 1867, effecting a pronounced drop in mortality and morbidity due to surgical sepsis. It was a milestone in the evolution of surgical practice from the era of laudable antiseptic surgery involved the use of carbolic acid. He is also known as the father of antiseptic surgery. Now let's know about another scientist named Robert Koch. In 18 he was from 1843 to 1910. Robert Koch was a German general practitioner. He is also known as the father of bacteriology. His contributions are uh, let's know about his contribution. First point is perfected bacteriological techniques and introduced methods for isolation of pure strains of bacteria. Second point is introduced methods of obtaining bacteria in pure cultures using solid media. Third point is introduced staining techniques. Fourth is discovered the anthrax bacillus, that is in 1876, tubercle bacillus, and the cholera vibrio, that is in 1883 he discovered. Fifth point is Koch postulates, that is, according to Koch postulate, a microorganism can be accepted as the causative agent of an infectious disease only if if it possesses some conditions. Let's know about the conditions. If it's fulfilling the conditions, first point is the organism should be constantly associated with the lessons of the disease. Second point is it should be possible to isolate the organism in pure culture from the lesson of the disease. Third point is the isolated organism when inoculated in suitable laboratory animals should produce a similar disease. Next point, it should be possible to re-isolate the organism in pure culture from the lessons produced in the experimental animals. Next point, these are the points. And an additional creation introduced subsequently requires that specific antibodies to the organism should be demonstrable in the serum of patients. These postulates have proved to be useful in confirming doubtful claims made regarding the causative agents of infectious diseases. However, it may not always be possible to satisfy all the postulates in every case. An important example of not fulfilling Koch's postulate is lepra bacillus. Sixth point, let's know about the about the contributions of Robert Koch. That is sixth point. Koch phenomenon. Sixth point is Koch phenomenon. That is Robert Koch observed that guinea pigs already infected with tubercle bacillus responded with an exaggerated inflammatory response when injected with the tubercle bacillus or its protein. This hypersensitivity reaction is called Koch phenomenon. Next point is Paul Ehrlich. It's a Paul Ehrlich from 1854 to 1915. Paul Ehrlich was a German scientist and is also known as father of chemotherapy. His contributions are we should know about some contributions. First point, uh, applied stain. First point is applied stains to cells and tissues for study of their functions. Second point, reported the acid fast nature of tubercle bacillus. Third point is proposed side chain theory of antibody production. Fourth point is discovered salvarsan, an arsenical compound sometimes called the magic bullet. It was capable of destroying the spirochete of syphilis. Later on, the discovered neosalvarsan and thus created a new branch of medicine known as chemotherapy. Fifth point is introduced methods of standardizing toxin and antitoxin. Now let's know about the virology. What do you mean by virology? The existence of viruses became evident during the closing years of 19th century when many infectious diseases had been proved to be caused by bacteria. Overall, what do you mean by virology? Virology means study of virus is called as virology. So, but there remained a large number of infectious diseases for which no bacterial cause could be established. During studies on rabies, Pasteur suspected that the causative agent could be too small to be seen under microscope. The existence of such ultramicroscopic microbes was proved when Ivanovsky in 1892 reproduced mosaic disease in tobacco plant by applying the juice of diseased plants to the healthy leaves. Two scientists, Loeffler and Froch, in 1898 observed that the causative agent of foot and mouth disease in cattle could also pass through a bacterial filter. The first human disease proved to be of viral origin was yellow fever. It was discovered by Walter Reed in 1902 in Cuba, and he established that the disease is transmitted through the bite of infected mosquitoes. Landsteiner and Popper in 1909 demonstrated that poliomyelitis was due to filterable virus, and they also transmitted the disease experimentally to monkeys. God Goodpasture in 1930 developed techniques of viral cultivation in chick embryos. The application of tissue culture expanded the scope of virological techniques considerably, and though the larger viruses could be seen under light microscope, after appropriate staining, detailed morphological study of viruses became possible only with the introduction of electron microscope by Ruska in 1934. The possibility of causing cancer was
first put forth by Elleman and Bang in 1908 in fall leukemia. Another scientist Ross in 1911 isolated a virus causing fall sarcoma. Several viruses have since been isolated which cause tumors in animals and birds. Viruses can also cause malignant transformation of infected cells in tissue cultures. The discovery of viral and cellular oncogenes was a breakthrough for possible mechanism of viral oncogenesis. The positive proof of viral causation of human malignancy was established when the human T-cell leukemia virus was isolated in 1980. The bacteriophages were discovered by Todd in England in the year 1915 and D. Harrell in 1917 in France. The molecular biology owes its origin largely to study on the genetics of bacteriophages and bacteria. Immunization of healthy individuals with dried cost of smallpox was a prevalent practice in India and China from sometimes immemorial. This was done to produce a mild form of smallpox, that is virulation. Though it was effective, it was hazardous. Edward Jenner accidentally came to know that the milkmaid who suffered from cowpox were immune to smallpox. Another scientist named Jenner in 1796 introduced the first successful immunization in smallpox by a related but mild live virus of cowpox. Now let's know about immunology. What do you mean by immunology? Boring and Kita Kita Sato Kitasato in 1890 produced antibody in guinea pigs serum by injecting a series of sublethal doses of tetanus toxin. The antibody so produced could neutralize the tetanus toxin specifically. The tetanus and diphtheria antitoxins were first known antibodies. Mm, Before in 1893 injected live Vibrio cholera into guinea pig which had been previously inoculated with killed cultures of Vibrio. This Vibrio these Vibrios were sown cultured of Vibrios. The Vibrios were shown to undergo lysis due to bactericidal effect. Bordet, another scientist named Bordet, in 1895 repeated the experimental experiments and demonstrated that two components of serum participated in lytic reaction, the first being heat stable and the second being heat labile. The specific antibody sensitized and vibrios and the lytic reaction was due to complement. Later on, agglutinin, precipitin, complement fixing antibody were demonstrated in serum. Metchnikoff in 1883 proposed that phagocytic response was the prime defense mechanism against microbial invasion of tissues and thus the cellular concept of immunity was established. Opsonin in serum was described by Dinsey and Lickliff in 1895 and Brett Douglas in 1903. In 1955, another scientist named Journey proposed the natural selection theory of antibody synthesis. Burnet modified this into the clonal selection theory. In 1967, Bonnet proposed the concept of immunological surveillance, according to which the primary function of the immune system is to preserve the integrity of the body, seeking and destroying all the foreign antigens. Malignancy was visualized by a failure of immunological surveillance. The scope of immunity was enlarged to include natural defense against cancer. Due to the work of Medawar and Barnett, it became possible to understand the immunological basis of transplantation. Successful transplants could be made possible by elective suppression and proper selection of donor on the basis of histocompatibility. Now let's know about chemotherapeutic agent. A scientist named Salvarson, an arsenical compound discovered by enriched 1906, sometimes called the magic bullet, was capable of destroying the spirochete of syphilis. He is also known as the father of chemotherapy. Gerhard Domag in 1800, from 1800 to 1895 to 1964, found that proton seal and azo dye was active against specific bacteria. In 1928, Sir Alexander Fleming accidentally discovered that a substance produced by mold inhibited a number of disease producing organisms. This substance was named penicillin, and this was the beginning of the antibiotic era. Other important antibiotics were discovered in rapid succession. The first success in treating streptococcal infections with sulfonamide was reported in 1935. A number of scientists in medicine and physiology have been awarded Nobel Prizes for their contributions in microbiology. Finally finished. Any questions regarding this? You can ask me in the comment box and subscribe my channel for more updates. You can know about biotechnological topics from my channel. Thank you.